So now I'll give a gentle introduction to polygenic scores, PGSs, or polygenic risk scores, which are special types of polygenic scores. So as you can see, they're kind of like having the risk of disease versus the reference group. It's about a risk of having a certain disease. That's what makes it a polygenic risk score. So to begin with, I'll be talking about SNPs, SNPs, single nucleotide polymorphism, SNP. Okay, so these SNPs, they're the most common type of genetic variation among people. So a single nucleotide, A, T, C, or G in the genome, in the DNA, it's altered in a specific chromosome and a specific position. So a DNA sequence might change from AAGCCTA to AAGCTTA. So for instance, it could just be different, like the C is replaced by a T here in that example. And you can see over here, for instance, that instead in one individual, it's an A in this position, and the other individual, it's a G here. That's a single nucleotide polymorphism. In this other one here, it's a C in this position, a cytosine, and in this other person, it's a T. And they're pretty common. So they occur approximately once every 300 nucleotides. So across the whole genome, there are roughly 10 million SNPs. And most SNPs have no effect on the health or development. Some SNPs are associated with diseases, with drug responses, or with traits. So for instance, if you were to take individuals who have a disease and individuals who don't have a disease, get their genomics information and find out where their genetic variants are relative to a reference genome, you could then find that some of these mutations, these variants, are associated with the disease more than others, and others might not be. So these are really important because some of these SNPs will be key for predicting someone's a risk for having a condition. So what is a polygenic score, a PGS? A polygenic score is a general score for any trait, like high intelligence. It could also just be a disease. Disease can also be a trait, like having Alzheimer's disease, or having schizophrenia, having heart disease, having certain cancer, right? So it's a genetic score for any trait. It's just in general. But a polygenic risk score will be a polygenic score that is focused on disease risk prediction. So you'll see that this is like low risk, this is high risk. It's like a bell curve. And a person's polygenic risk score is a statistical calculation that is based on the presence or absence of multiple genomic variants without taking environmental or other factors into account. So it's a single number that basically encapsulates and summarizes an individual's genetic predisposition to a trait or disease. And it's computed as the weighted sum of risk alleles across many genetic variants like single nucleotide polymorphisms, SNPs. So we find risk SNPs for a given condition, and we can use those to see if an individual has those risk SNPs, and what are the dosages. Do they have them on both the chromosomes from both parents, from just one parent, or from neither? Kind of sum up those risk variants, and then find out what could be their likely score for having the condition. And if it's a PRS, which is what we're looking at in this example, even though PGS calc can work for all sorts of traits, we're just looking at a disease risk prediction for late onset Alzheimer's disease in this tutorial video. So it'll be a polygenic risk score. So that's just a specific type of polygenic score. And many traits like height, body mass index, diabetes, schizophrenia are polygenic. They're influenced by thousands of SNPs. That also makes it very challenging to predict the etiology, the start, and what's causing the disease, right? It's very difficult. And that's why there's a huge need for precision medicine. There are thousands of SNPs that can be contributing to these traits that we observe. Why are certain people taller than others, and so on. And a polygenic score captures the combined small effects of many variants. So it's useful for risk prediction, for stratification of individuals, putting them into different groups, and for personalized medicine, also known as precision medicine. So it's a great day to score above the curve. Well, maybe if it's a polygenic score. I'm not sure if it's such a good idea. If it's for, disease prediction, I'm not sure. Probably not. You want to be under the curve, SpongeBob. So, as I was saying, a polygenic score, it's a number that summarizes a person's likelihood of having a given trait. And if it's a polygenic risk score, it's a number that summarizes a person's genetic risk for a trait or a disease. So think of it like a credit score for your genetics. The higher or lower the score, the more or less genetic risk you may carry. So how is it made? Scientists take results from large genetic studies like genome-wide association studies, where you have individuals and then, you have genotype them, and you find the individuals who have a condition and individuals who don't have a condition, and then you try to associate and find which are the regions in the genome where the differences are. You kind of align them to a reference genome, and then you see, looks like on this position, at this point, a lot of people who have that base instead of another base, if they have a G instead of a T, it seems that those individuals have a high likelihood of having the condition. So that could be like a risk factor from a genome-wide association study. So, we take these results from large genetic studies, genome-wide association studies, GWAS, and each genetic variant gets a small weight based on how much it influences a trait, and they are added up across thousands of variants to give an overall score here. So, a polygenic score is not one gene equals one disease. Instead, it's many small effects added together. So it's advanced analysis of our DNA beyond one SNP at a time. So you can't just say, you have this, like for breast cancer, it's usually the BRCA1 gene, BRCA2 gene. Those have high risk if something goes wrong with them. But as you can see here, and this is what these SNPs usually look like. They have these RSIDs, reference SNP IDs, in front of them. It usually corresponds to a chromosome number and a position and a base change, and like instead of having a C in a spot, you may have a G or something, for instance. So it's like a credit score, right? You have these, this is your genotype, this is your score, and then, you kind of compute what this is. And you can kind of find this genetic risk score. And these small effects, they come from, these SNPs, this single nucleotide polymorphism, the tiny differences in DNA between people. Like, we're remarkably very similar to these monkeys and so on. So a lot of the DNA is shared evolutionarily. So a lot of us share a lot of the DNA. It's just this few bases that make us very different. So for instance, most people could have an A at a certain spot, but some have a G, and that's a SNP. So kind of taking somebody's genotype, finding what SNPs they have, and then we have like models that kind of associate, with, certain SNPs with the disease, and then we can kind of predict somebody's risk score for having it given their genotype. And that is what precision medicine fundamentally is, taking somebody's genotype and coming up with a predictive number of how likely they are to have a certain condition. So as I was mentioning, SNPs, they are a single base pair change. This is a reference SNP ID in the DNA sequence. So over here, you can see that at this particular position in the DNA, at this chromosome number and position, a person has a C over here. Kind of just zooming in along, this is the DNA over here. The DNA, if we're unwinding it, these are histone proteins, we're unwinding it, and we're looking inside the nucleus of a cell, and we're seeing the DNA across the chromosomes, and then we can see that this cell belongs to a certain tissue, and in the human, for instance, and this SNP could be why an individual could be affected for a disease, for instance. If we do a genome-wide association study and look at the Manhattan plot over here, what we can see is that this SNP at this chromosome position 6, at this spot here, it's above the genome-wide association threshold of significance, which is usually 5 times 10 to the negative 8, because given that we're testing millions of SNPs. So this SNP is strongly associated with having increased risk of the disease. of being affected, in people who have this SNP. So yeah, they can be common with a minor allele frequency greater than 5% or rare, usually a biallelic, two possible alleles at one position. And the importance is that some SNPs affect gene function or regulation. They can be coding found right within a gene itself. Genes are along the DNA, codes for genes. The RNA polymerase 2 makes a transcript of it. Messenger RNA leaves the nucleus, gets translated by the ribosome into proteins, which do a lot of functions, right? So other SNPs are neutral markers, but they can still track inheritance. So it's used as genetic markers to find associations with traits or diseases. So yeah, these genome-wide association studies, they test millions of SNPs for association with phenotypes like the diseases over here. And there can be like linkage disequilibrium mapping to identify regions of the genome related to the disease. Functional studies like SNPs may disrupt protein function or regulation. And population genetics, they can be used to study ancestry, migration, and genetic diversity. So SNPs are the fundamental units of genetic variation, and they form the basis for studying the genetics of complex traits and diseases. So in this slide, you can see, the genotype here to the phenotype links over here. And they are the building blocks of the genetic analysis in various data cohorts. All of us, I'm a participant in that actually, UK Biobank, Our Future Health, so on. So yeah, this SNP is associated with DNA base C at this position, and it's strongly associated with the disease based on this Manhattan plot. And there can be various analyses to determine the impact of this disease risk SNP on an organism. For instance, there's expression quantitative trait loci, EQTLs, that associate SNPs with changes in gene expression. So SNPs can impact phenotypes in various conditions that we observe. Basically, it's what are the differences between individuals that are pretty common. So single nucleotide polymorphisms, it's a one nucleotide difference in a particular position of the deoxyribonucleic acid of the DNA in a specific position. They're the most common type of genetic variation in humans. And so this Manhattan plot here, because it looks like the Manhattan, New York City skyline, that's why it's called Manhattan plot. It's from this recent genome-wide association study, GWAS, for Alzheimer's disease. And so what we can see is, this is genome-wide association study. We have a genome-wide significance threshold, a p-value of less than 5 times 10 to the negative 8, because we are testing millions of SNPs, so it's a pretty strict value. And you can see along here going across the chromosomes. And these are the negative log 10 p-values here for the association analysis of this SNP's association with Alzheimer's disease risk in cases, people with Alzheimer's disease versus controls. And as of now, this peak here corresponds to ApoE4, so the E4 version of the apolylipoprotein, has E2, E3, and E4 alleles. And the E4 allele here, it's the only known causal determinant for late onset AD, the largest known risk factor for late onset Alzheimer's disease. A lot is unknown, and researchers are still trying to figure out what are risk factors, SNPs, and variants for Alzheimer's disease. But in general, polygenic scores and polygenic risk scores, PRSs, they estimate an individual's genetic predisposition to a trait. That's what polygenic scores do, or a disease. So polygenic risk scores are polygenic scores that are focused on diseases. Because the trait can be like intelligence, high, it could be something positive like a polygenic score. But a polygenic risk score is a polygenic score that is applied towards a disease. And so basically what these scores are trying to measure is how likely is the individual to develop a certain trait or a disease, based on their genetics, based on, whether they have certain SNPs or not. And it's calculated by combining the effect of many genetic variants like SNPs across the genome, across all the chromosomes, in our cells. So how they calculated for the polygenic risk score, we have the effect size of a SNP and then the number of risk alleles. So for each SNP version that you have, what is its effect size on the disease or the trait? In this case, the disease, because it's a PRS, but same thing for polygenic score. So what is the effect size multiplied by the number of risk alleles, like the number of alleles that you have that are of the risk allele. So I will show you an example, but this is sort of based on data from the GWAS, genome-wide association study, and it can include hundreds to millions of SNPs, and I'll show you a very toy example, but that's real, that's just 21 genetic variants, but you basically take for each SNP, its effect size, for a given individual. If it has an effect size of that SNP times how many risk alleles the individual has for that SNP. And then you keep on, it's a product, and then you just keep on multiplying, you keep on summing up these products, multiplying the effect size of a SNP for another SNP times the number of risk alleles for that SNP. Then it's like a weighted sum, it's a sum product here. So yeah, a SNP, a single letter change in the DNA sequence, most common type of genetic variation. Most SNPs don't cause disease directly, but when you study millions of them, some are associated with traits like height, cholesterol levels, disease risk. SNPs are like spelling differences in a book. Most people don't change the story, but some can change the meaning or give clues about the plot, So I'm from the US and I moved to the UK recently, and I can see that they spell things a little bit differently, like realized. That's something I realized, is that I spell it REALIZED and they spell it REALISED, realized different, right? So it's a SNP in my definition, I guess. So, this Manhattan plot is actually from Alzheimer's disease, a 2021 study here, with so many cases, people with the disease, and so many controls. And researchers use a variety of metrics to link disease risk SNPs to disease genes. And ApoE4 is the only known causal determinant for late onset Alzheimer's disease. A lot of Alzheimer's disease metrics are still unknown. So linking back to polygenic scores, a polygenic score is built from lots of SNPs. Each SNP contributes a little bit, positive or negative, to the final score. Together they create genetic risk estimate for complex traits. And so a lot of the genetics remains unknown about Alzheimer's disease, but polygenic scores and polygenic risk scores, they estimate an individual's genetic predisposition to a trait if it's a PGS, or a disease if it's a polygenic risk score. So how likely is the individual to develop a certain trait or disease, and it's calculated by combining the effects of many genetic variants across the genome. So we have an effect size of the SNP from a genome-wide association study, and we multiply that by the number of risk alleles that the individual has, and it's based on data from genome-wide association studies that can include hundreds to millions of SNPs. So an analogy from one of my favorite shows of all time, SpongeBob SquarePants, is that a polygenic score is like a recipe, for making Krabby Patties, pinch of salt, what are the different things that go into it. So, and the point is that as part of this recipe, this product of Krabby Patty, to make that you have certain ingredients that come in. I'm sure like eggs don't just turn right into Krabby Patties, but you need a certain set of ingredients to make them, and to follow that recipe, the ingredients are SNPs. SNPs are what kind of makes the final polygenic risk score. The ingredients are what goes into a Krabby Patty. So similarly, the analogy is that a polygenic score is the recipe and whatever we're trying to make, whether it's a Krabby Patty, like Patrick is devouring it so much, and the SpongeBob, he's looking at Krusty Krab making the Krabby Patties, and the single nucleotide polymorphisms, the SNPs, are the ingredients that go into that Krabby Patty. So what goes into making a polygenic score are the SNPs. They're the building blocks of that score. So the recipe is a polygenic score, and it's kind of made up of a bunch of these ingredients, which are the SNPs here. A SNP specified here, for what that position and that base pair. and what you have. The XIJ is a genotype for the ith individual, and the jth SNP, and it can either be a 0, 1, or 2 for the effect allele dosage, and I'll explain that next. But basically what this beta is it's the effect size of that SNP from like a genome-wide association study analysis and so on. So what you do is, for those risk SNPs, you basically kind of find out, if the individual has those, like if it has that genotype for that risk SNP, and you basically are summing across the risk SNPs and how much each SNP is supposed to contribute to the disease. So the SNPs are the tiny building blocks of genetic differences, and these genetic scores, they're the summary score we get by adding up the effects of many SNPs. And if this doesn't make sense, I'll be walking you through an example. But each polygenic score is calculated as the linear sums of the effects of these M variants. You're summing up these M genetic risk SNPs, based on the effect weight in the scoring file, beta. With this XIJ is this genotype for the ith individual and jth SNP, and this genotype is usually 0, 1, or 2. 0 means that you don't have both of the effect alleles. 1 means that you have one from one parent and one from the other. You have one effect and then one non-effect allele. And then 2 means you have both of the effect alleles, and the effect alleles are the ones that are considered to be contributing to the condition. So I'm going to show you an example of how we calculate, in this case, a polygenic risk score for disease. It's just a polygenetic score, but here it's a disease. So let's just see this example. So RSID, these are SNPs, reference SNP IDs. So we have a score file that has J equals 3 SNPs that contribute to the disease. So there are 3 SNPs that are considered to be contributing to the disease. This one, and this is just a toy example, so it's on chromosome 1 at this position here, and the effect allele is an A, and usually we also would have another allele, like a non-effect allele, but just for simplicity I've put the effect allele here, it's an A. And for the disease, it's considered to have this weight, this is the weight that it contributes towards the sum, towards the polygenic risk score. And this is this other SNP on chromosome 2. So these other 2 are on chromosome 2 at these different positions, and they're both a T over here. So you can see this one has the largest contribution to the disease, and this one has the smaller contribution, the smallest to the disease. And then we have the genotypes of 2 individuals, so the count of the effect allele. So 0 in the genotype beta means that you have both non-effect alleles from your parents. That means that you don't have this allele of an A, for instance, or so on. And 1 means from both parents. If you have a 0 for the genotype, then it's from both parents you don't have the effect allele. So for instance, what we see over here is that for these 3 SNPs and these 2 individuals, we see the individual one's genotype is a 2, which means that for this SNP over here, they have both effect alleles of AA from each parent. So for this parent, for this guy, it would be an A and an A for this person here. A and A for this point here, for this person. It would be an AA over here. Sorry, that came out a little bit messy, but the thing is, it would be an AA here. And then for this individual, they have a 1. The same person has a 1 for this SNP here. So it means that they would have a T from one parent and the other non-effect allele from the other parent. And they have a 0 for this one, which means that for this SNP, they actually do not have this effect allele T for, in their genome. They don't have it at this chromosome 2 at this position. They don't have the T on either of their chromosomes, because we have one set from our maternal from our mothers, biological mothers, and one set from our biological fathers. So that means that they don't have the effect allele. So how many of the effect alleles do they have? And usually we would know what the non-effect allele is, but it's not mentioned in this example just for simplicity. Whereas for this individual, they have a 0 here for this SNP. So that means that for both Parents, they do not have an A at chromosome 1 at this position here. And similar to the first individual, they have like one of the T alleles at this position from one parent, and from the other parent, they do not have that. They have the other one. And lastly, for this one, it means that for this individual at chromosome 2, at the position 234,068,476, they have a TT at that spot, one from the biological mother, one from the biological father. So this is the genotype data that we have. And this is the polygenic risk score calculation formula. So it's basically saying that these are the weights that each of the different SNPs contribute towards the disease or the condition. If it's a polygenic score, it's the same formula, but this is just like any trait. It can be a disease or not. So a polygenic risk score is just a specific type of polygenic score that is focused on a disease. That's what it is. It's just a specific type. And since here I'm just showing a disease, it's a PRS. The same thing is for a PGS in general. It's the same thing. It's just that we're measuring some trait in general. That's what a polygenic score is. So all we're trying to do is these are basically counting the number of effect alleles that we have. And we know that for every effect allele, this is you have from each parent, this is how much it contributes to the disease. So basically, what we are doing then is we're combining the two tables into one. We can do that. So for the SNP, we have the effect allele, the weight, and the individual's one's genotype and individual two's genotype. And all we're doing is this is just telling us for every effect allele that you have of this type, for this SNP here, if you have two of them, you multiply it by 2. If you've got one of them, you multiply this by 1. If you've got none, you just leave it at 0. So you're basically doing for this individual one, based on this formula, you're taking the genotype of the individual and you're multiplying it by the effect weight beta. So for individual 1, for this SNP, here we're just taking the 2 and we're multiplying it by the 0.166 and we're getting a 0.332. That's what we're getting here. That's how much they have based on this SNP and its importance and their genotype. For the second SNP, we take this 1 and then we multiply it by this 0.199 because they just have one copy of this from one parent. If they had a 2 here, it would be multiplying this by 2. So we just take the genotype here and we multiply it by the effect weight. So this times this. And we'll note that it's the same for individual 2 because they also have a same effect weight. So it's the same thing here, 0.11 and a 0.199. And lastly, for this person, since they don't have this effect, they don't have this particular effect alleles, it's just going to be 0. So no contribution of this SNP to their risk score. So it's a 0 here. And all we do is we just sum them up. So this plus this plus this is equal to 0.531. Similarly, for the second individual, we take this SNP here, multiply it by 0. They don't have either of the effect alleles from either parent. So it's a 0, no contribution from this SNP. Again, this is 0.199. And this one here is just a 2 multiplied by this value here. They have both effect alleles and for this SNP, and this SNP has this contribution to the disease. So it's 2 times that. And we add this up. And you can see that it's a 0.353. So this is individual 1's polygenic risk score for the disease. It's 0.531, which is higher than individual 2's polygenic risk score for the disease, which is 0.353. So individual 1 has a higher risk of the disease, a predicted risk, than individual 2 does based on this toy example. So that's how we compute a polygenic risk score. We take the genotypes respectively and multiply by for that given SNP, and we multiply it by the respective weight. And the genotype is just measuring how many copies of that effect allele do you have, that risk allele or so. So yeah, basically, each SNP has an effect size from a genome-wide association study. The individual's genotype at each SNP is multiplied by that weight, the effect size, that contribution of that SNP. That's usually given by that beta, that weight of that SNP for the disease. And then, if it has a higher weight, it's a bigger contributor to the disease. And then you add up across the SNPs those weights, and then you get a polygenic score. And if it's for the disease, it's a polygenic risk score. Otherwise, if that score is just like height, intelligence, body mass index, and so on. And, the traits can be so various. It can be, are you a morning person or not? Like your likelihood of being a morning person, having insomnia, being risky or risk averse, right? So when we compute the polygenic risk scores, we can kind of put them over here, and this is like a standard z-score where, in the middle it's like a bell curve where it's like, oh, I cannot draw. Let me show you, for instance. It's just going to be, and this would be like a 0 and like a 1, a 2, standard deviations away from the mean, and a 3, so it's accented at 0 if it's a z-score, and so on. And then on the other side it would be like -1, and so on. It's just like -1, -2, -3. And you can see that, if an individual is in the 95th percentile, they're in the highest genetic risk group, based on their z-score. They are extremely high predicted risk for having a certain condition, like in the disease in particular. So yeah, this is just a toy example for how we can compute like a polygenic score or a polygenic risk score in the case that this is a disease, for instance. But we're just going to be expanding on this idea using the PGS catalog calculator for real-world disease or a real-world trait, and kind of how we can use that to compute this. It's sort of, this is the kind of like the idea behind the scenes on a very simple level, and then from there it's just a little bit more complicated with the real-world data, with a lot more SNPs and different genotypes aligning strands and so on. But this is just the general idea as well of how you can compute a score for the disease, and we're going to be running this using the PGS catalog calculator tool. So to compute these polygenic scores, we'll be using the polygenic score catalog PGSc calculator tool here. So this is the workflow. We'll be using this function here. This is the GitHub here. And it's based on this recent paper published in Nature Genetics called Enhancing the Polygenic Score Catalog with Tools for Score Calculation and Ancestry Normalization. So it says that these PGS scores have transformed human genetic research and have numerous potential clinical applications. So they present recent enhancements to the polygenic score catalog and highlight this calculator. It's an open source, scalable, portable pipeline for reproducibly calculating PGSs. So it's useful across a lot of applications, and we'll be using that here in this example to compute the polygenic risk score of an individual developing late onset Alzheimer's disease. And currently, there's no tool in the UK Biobank for using the polygenic score catalog calculator. So I had to find a workaround, which I'm showing in this video, for how we can run this tool using the DNA Nexus platform. And this can also be a guide just in case you have other tools that you want to run that are not readily available in the DNA Nexus UK Biobank platform. This is sort of like how you can run those tools exactly, like how you can download them, how you can run them in the DNA Nexus platform. So even if you're not interested in running PGS catalog calculator, you can kind of use these concepts, hopefully, and the example codes to extrapolate and see how you could run your codes, potentially. So I hope it can help you. So you might be wondering, why not use Plink2//score, the tool? And we do use Plink2 in this video for other things, so it's no hate against Plink2 at all. because we do use it for other things in this video, but it's very powerful of a tool. It's a very powerful tool. But you might be curious what PGSC Calc does that Plink 2 Score doesn't, and so they've written this part in the PGSC catalog calculator tutorial part, and they say that we use Plink 2 internally to calculate all scores, but offer some extra features. So this is what they've mentioned here that they do that is a bit more advanced than what Plink 2 does. So they match the variants from scoring files, often from different genome builds, against the target genome using multiple strategies, taking into account the strand alignment, ambiguous or multi-allelic matches, duplicated variants, matches, and overlaps between datasets with a reference for ancestry. So if reference and alternate alleles are mixed around across datasets, or the strands are different, the DNA strands, it does all the matching. If there's flips and whatnot, and in other types of inversions and DNA strands, it handles that. One important output is an auditable log that covers the union of all variants across the scoring files, tracking how the variants match the target genomes and provide reasons why they are excluded from contributing to the final calculated scores based on user-specified settings and thresholds for variant matching. From the matched variants, the workflow outputs a new set of scoring files, which are used by Plink 2 for scoring. The new scoring files combine multi-polygenic score in a single file to calculate scores in parallel. These scoring files are automatically split by effect type across duplicate variant IDs with different effect alleles. Then the split scores are aggregated. The pipeline also calculates genetic ancestry using a reference panel. The default is 1,000 genomes, handling the data handling, variant matching, derivation of the principal components analysis space, and projection of target samples into the PCA space using robust methods. So they implemented here. And scores can be adjusted using genetic ancestry data using multiple methods. So they have this here. So they have a summary of all the benefits here of why we should use it. And I will be computing polygenic scores using the polygenic score catalog calculator going forward. But yeah, Plink 2 is also great, but this is sort of building on it. So